So our next speaker is Danielle Bassett from the University of Pennsylvania. Now from bacteria to human brain, neuroscience.
Great, thank you. I wanted to thank the organizers for inviting me. This is a very interesting meeting. I'm going to be talking about network dynamics and control in the human brain, so I've changed my title slightly, and I'm going to expand some of the ideas that I was initially planning to talk about. So this talk will be of a flavor that's very embedded in the application, so I'm hoping that as I talk about this work, you'll learn something about the brain as well. So just to begin, I'd like to say that there's an emerging view of the brain as a networked system, and that's over a variety of different spatial scales as well as a variety of different ways of measuring connectivity in the brain. So on the top left here, what I'm showing you is a pattern of structural connections inside of the brain. So just to give you an idea of what you're looking at, this is the front of the brain, this is the back of the brain, and this is the top, and this is the bottom. And these are actually wires that are inside of your head, which are bundles of neuronal axons. So those are pathways along which information, and particularly electrical signals, can be passed in the brain at the very, very large scale. In addition to that, there are also networks at other spatial scales at the level of very, very tiny volumes of tissue all the way up through large volumes of tissue. And this emerging view of the brain as a network system impacts on many of the questions that neuroscientists would want to address. And one of the questions that I think is very paramount in many people's minds right now in trying to understand the brain is the question of control. And this network nature of the brain inherently changes how we think about control. So just to be very clear about what I mean by control, there are two types of control that we might wish to understand. The first is internal control. So this is, how does the brain actually control itself? How does it control its own patterns of activity? This would include ideas like cognitive control and homeostasis. On the right-hand side, I have external control. So this is, how do I intervene, or how does a clinician intervene to change the direction of brain activity, particularly in individuals who have neurological disorders or psychiatric disease. And this external control can take the form of, let's say, stimulation. So here, for example, is a picture of transcranial magnetic stimulation, which is the use of a magnetic wand to inject electrical current into the brain to change the activity of a particular brain area. This is commonly now being used to treat major depressive disorder with some success. So it's something that is very important right now on the clinical front. What I'm going to focus today's talk on is cognitive control and trying to understand dynamic networks in the context of cognitive control. So what is cognitive control? If you were to ask a neuroscientist what cognitive control was, this is the answer that you would most likely get. So cognitive control is a term that's often used to describe attentional control and inhibitory control. It's implicated in cognitive processes from working memory, cognitive flexibility, reasoning, problem-solving, planning. What we often think of is that it's the ability of the brain to switch between different tasks. So the ability to switch between, let's say, solving a mathematics or statistics problem and talking to your 2-year-old or to your grandmother, right? Sometimes these transitions are very easy for some of us. They're very difficult for others of us. I am one of the people for whom transitions like that are actually quite hard. This is also the ability to inhibit inappropriate behavioral responses. So to stop yourself from doing something that's inappropriate socially or emotionally. And then to link multiple sources of information to solve problems. So how do we study cognitive control, and how do we understand how the brain actually does it? One of the ways that we try to study cognitive control is by building a functional graph from data taken from the brain. So what we do is that we subdivide the brain into distinct areas that are normally based on anatomical boundaries. So there are some parts of your brain that have a specific distribution of neurons or cell types, and another part of your brain may have a very different distribution. And so there are sort of natural boundaries between areas that you can identify based on the anatomy. So those are the nodes inside of our network. The edges that we'll be talking about for this part of the talk are based on coherence between signal time series. So when we measure...
So you're looking at the actual neurons, this is not like fMRI or something?
This particular slide is just sort of a general idea of if we have neuroimaging data and can measure either activity of neurons or of large-scale brain areas, like an fMRI, this is the way that we might construct a graph. The next slide will actually be fMRI data. So what we'll do is that we'll connect up those individual brain regions using the coherence of activity time series from the areas. What do we see when we do this in fMRI, particularly when individuals are just sitting resting inside of an MRI scanner? One of the really salient and I think striking structures in these networks is community structure. So what you can observe here is these are dots, so regions of interest that are placed on the surface of the brain. Here's the left-hand side, the right-hand side, and these two are the medial wall, which is the inside of the brain, the sort of sides of the two hemispheres. On the left-hand side is the functional network that's been reconstructed from time series extracted from those areas. So you can see that there's really beautiful clustering structure. There are regions, these blue regions here are more densely interconnected to one another than they are to the red regions here. So that was reported back in about 2008. And since then, what people have realized is that each of these little clusters maps really well onto things that your brain does, to say it quite colloquially. So regions that are really important in moving your body all seem to show very strong functional connectivity with one another. Regions that are important in vision are all connected to one another. Regions that are important in helping you make difficult decisions or helping you remember something are all densely connected to one another. So if you look at the community structure inside of these functional graphs, you can learn something about the fundamental processes that the brain performs for you in your everyday life. But which of these little systems or subsystems is important for cognitive control? There are three different subsystems that are important for cognitive control. And they are called the default mode, the frontal parietal, and the attention circuits. I'm going to map the regions that are important for those three little subnetworks onto the surface of the brain here. So blue is the default mode, green is the frontal parietal, and red is the attention. And what I want to sort of say is that, while it's interesting to identify which parts of the brain are important for cognitive control, it doesn't necessarily tell you how that happens, right? Knowing where in the brain does not amount to knowing how in the brain. So what I've been trying to do is to ask the question of how those regions actually support cognitive control. When you're asking the question of how, you're asking a question of dynamics, right? You're not asking a question of location. So what we'd like to do is to understand the dynamics that support cognitive control. So from neuroimaging data, what we do is that we take the brain, we subdivide it into different pieces that we're interested in. We extract activity from each of those areas, let's say using fMRI or other types of imaging techniques. And then often what we do is that we'll separate that data into little time windows. So here's one time window. I'll construct a coherence-based network from that time window. Here's another time window. I'll construct another network, another, and another. I've given an example of 4 networks in a row, but from neuroimaging data we can construct, if we have fMRI, we construct a couple hundred networks in a row. If we have higher resolution, temporal resolution imaging data like EEG, for example, or electrocorticography, then we can make many more, thousands or more, depending on the length of the acquisition. Okay, and so then what do we do with that data? Obviously that's one sort of coarse representation of the data, but it's still actually fairly complex. So one of the questions that we've been very interested in is how does that modular structure or community structure that we observe at the summary level, how does that change as a function of time as people are actually using cognitive control to perform different functions? So one of the techniques that we've been using is maximization of a modularity quality function that's been extended to multi-layer networks. And this has been in collaboration with Peter Muka and Mason Porter since about 2010, I think is when I met them. So it's been a really fun couple years of working on some of these problems. In the multi-layer approach, what we do is that we connect up a brain region in one time slice to itself in the next time slice. So instead of having an ordered set of matrices, now we actually have a set of matrices that are connected to one another by identity links. So I'm going to connect up node I in time slice 1 with node I in time slice 2, and then with node I in time slice 3. So instead of having independent adjacency matrices or even ordered adjacency matrices, I actually have linked adjacency matrices. And then we maximize this modularity equality function. Here is my adjacency matrix, which is the connection between, each element gives me the connection between node I and node J in the layer L, right? So now it's a three-dimensional structure, yeah.
So you're working at first with associations, and then you're thresholding those associations so that you have not a weighted network, but just
I'm not thresholding, no. All of the weights are still in there.
Oh, so you're
It's a
working
fully
with
weighted adjacency matrix.
Okay, so you're doing this as well with the weighted network.
With fully weighted, yeah. Yeah, and we wrote a methods paper, which is here, which was through some of the questions like that about, you know, implications of thresholding versus not, and different sorts of null models. Null models are very important because what this modularity maximization is doing is that it's trying to maximize the difference between the adjacency matrix and what's expected in a random network null model. Often what's used here is a configuration model, which many of us are familiar with, but in the case of spatially embedded networks or in the case of matrices that are built from either correlation or from coherence, there can be other types of null models that are particularly relevant. And those are questions that we started to address in this paper, and many others have been working on similar types of problems. But what this has done for us is that it enables us to identify what the modules are in a particular time slice and then how those modules change over time. When we study this, there's one particular parameter of that reconfiguration that we have found to be very, very related to how people behave. And therefore we think is very important for answering questions in neuroscience. What you are seeing here is, again, 4 networks in a row, and I want you to focus on this top region here. So this node is initially connected to the peach module, but then it becomes connected to the yellow module, and then becomes more strongly connected to the yellow module, and then eventually is extremely strongly connected to the yellow module. I see where the confusion came from. My schematics are binary graphs. I apologize. So these are, the actual data underneath of this is completely weighted, but sometimes the binarization helps you to get across some of the ideas a little bit more simply. So this change of a pink region affiliated with the pink module to the yellow module is something that we quantified using a notion called flexibility. So flexibility is the number of times that a node changes its module allegiance over time. So you simply count up the number of changes that a node makes to different modules over time, and then you normalize that by the number of changes that were possible in. Okay, so that's dependent on T, which is the number of matrices in your ensemble. So what we've done is that we've found that that measure of flexibility is something that changes across your brain. So there's some parts of your brain that are very, very flexible, and then there are other parts of your brain that are much less flexible. It also changes in different people. So some people have very flexible brains, while other people have less flexible brains in this specific definition. We've found that that amount of flexibility is correlated with how much somebody can learn a new visual motor skill. So for example, playing the piano or juggling, so learning how to use your eyes and your hands to perform a new function. It's also very tightly correlated with cognitive flexibility. So what is cognitive flexibility? So that's a notion from psychology, not from network science. Cognitive flexibility is the ability to switch between different strategies or rules to perform a function. And the common way in which we can test for cognitive flexibility is that we give everybody a piece of paper that has the numbers 1 through 26 randomly distributed throughout the page, and then it also has the letters A through Z randomly distributed throughout the page. And then we ask you, can you first find number 1, and then find letter A, then find number 2, and then find letter B, then find number 3, and then find letter C, etc., right? So you're going through numbers and letters in order, but you have to switch back and forth between, am I looking for the next number, or am I looking for the next letter? And the quicker that you can perform that whole task, so getting to Z and 26, is your measure of cognitive flexibility. Lower is better, right? The faster you can do that, the better you are at cognitive flexibility. That's actually something that has also been positively correlated with later success in life, including sort of how much money you make and your general happiness. So very, very interesting, something so simple. Yeah, go ahead.
Wouldn't you think that you could train for this task?
that you could train for it.
Right, and you can get faster and faster.
Yes, the big.
What can you do? You're an adult.
Most people don't do
that. You get free.
I think that you might be able to train for the task and get a little bit quicker. There's probably a ceiling on it. It's not something that you can just train forever. And the big question actually is generalization. So if you train on this task, does it generalize to other good things in your life? And that's a really, really big question. There's actually very little evidence for the generalization of executive function training, which is exactly what you're yeah, go ahead.
In the procedure that you're using, so you're doing community detection, I guess every node is associated with one and only one community, is that?
Right, so it's a hard partitioning algorithm. Yes, so when we maximize the modular equality function, we identify a hard partition of nodes into communities. What we do though is that because maximizing this equation is NP hard, we use heuristics to optimize it, and then we take an ensemble of near optimal solutions, and then we look at structures that are consistent over that ensemble. So in some case you can say if you're looking at the set of partitions, it's a little bit like understanding fuzzy clustering, but it's not exactly fuzzy clustering. It's based on an ensemble of hard partitions.
I was wondering how important background is. This is something that Peter and I have discussed, and the important, I don't, and it may not, in some networks, clearly there's a lot of nodes that are really not preferentially attached to anything. And that you may benefit from ignoring them. I don't know if this is the case where everything is kind of doing something important, or if it would be profitable to ignore a lot of areas, regions, nodes, voxels.
Right. We haven't usually ignored them a priori, but what we have done is that after we do the community detection, sometimes there are communities that just tend to be full of regions that were not very strongly connected to anything. And then we do a non-parametric permutation test to say that particular community is not enriched for strong connections any more than expected. And so therefore we can ignore it. It's a great question. Okay, we also find that this flexibility is related to working memory, so that's whether you can keep information online for a period of time, and other types of learning. So what all of these studies have basically done for me is that they've motivated the idea that cognitive control may be an explicitly network level process. And if that's true, then because all of these are very heavily dependent upon cognitive control. If that's really true, then what we'd like to understand is how these dynamics, these reconfiguration patterns, happen on top of the structural network that sits inside of your brain, right? So when we're talking about dynamic networks, obviously there are this dichotomy we always separate, which is the dynamics of the networks themselves, but also the dynamics that occur on top of the network. So what I'm going to talk about right now is how do the dynamics occur on top of that network.
The graphs are directed or undirected?
They're undirected.
Undirected. I mean, you would imagine that some region would trigger activity in another region.
Yeah, that's a great question. So the structural connectivity here, the way in which we measure it is it's based on watching the diffusion of water molecules inside of the brain. And our measurements of diffusion are not directional. So we can see kind of the axis of diffusion, but we can't see the movement of water molecules exactly. So we can't, and we can't see the flow of information directionally.
What's the time scale? I mean, diffusion perhaps is a slower process.
It is slow, and it's yeah, right. It's not. We are not currently confident using the imaging technique that we have to estimate directionality. If you have, however, this is for structure. For the functional networks, so when you take two regions and you connect them based on how similar their activity is, you could define a causal relationship there if you have enough data to be certain that your estimate of causality is true, right? So that's often done with very high temporal resolution data like in EEG or ECOG. It's less commonly done with fMRI data, particularly in these short time windows. Okay, so how does the actual structure support these dynamics that are occurring and that are helping you to perform the functions that are required by you in everyday life? So to answer this question, we've been thinking a lot about this field of structural controllability, which was developed in the 1970s. And the idea of structural controllability is basically that if you have a network architecture and you have the capability of injecting energy into that network architecture, you intuitively would know that if you inject energy into this orange point here, you're going to have a very different impact on the dynamics of the network than if you inject energy into this gray region over here, right? So if you inject the energy into this orange region, it's going to flow to all of these little modules very quickly, whereas if you inject energy into the gray region, it might get stuck in this little area for longer, right, and not impact the entire system. So the field of structural controllability gives you some ideas about how you would address that kind of question for any networked system. So we've been asking the question of how do we think about that in the brain? So if we have the structural connectivity of the brain, we know we have an adjacency matrix which is fully weighted, which tells us how each of these pieces are connected up with one another. Then we can ask the question, what is the amount of control energy that I should give to which nodes as a function of time in order to have this happen? So this is our outcome measure. We'd like to have the yellow region highlighted here increase in its activity over time. We'd like to have the blue region here first increase and then decrease in its activity over time. And then we'd like to have the peach region here decrease exponentially in its activity over time. So the big question is, given the structure of the brain, the structure of the network inside the brain, how would we enact this particular type of control? So in order to do that, what we really need is to map the actual dynamics that we anticipate to happen on top of the network. What we've started with is the very simplest model that we could come up with, which has also been suggested to be somewhat valid in very slow timescale neuroimaging data. And that is this noise-free linear discrete time and time invariant network model. So here what I have is at the x at time t plus 1. That's the state of brain regions at time t plus 1, where state is a measurement of activity over all nodes in the network. So it's basically a vector, which gives me the activation magnitude of all nodes inside of the network. I'm going to say that that is going to be equal to my adjacency matrix, which is given by how those regions are actually wired up structurally in the brain, times my state at time t, and then plus my control. So this is the use of kappa as the control energy that's going to be injected into kappa nodes at a particular time t, or over all times t. So this is my relatively simple model. And this is actually the model that most results in network control theory have been derived for, because it's the most analytically tractable. So the very first question that many engineers would actually ask if you talk about network controllability, they would say, well, is a system absolutely controllable? Meaning, can I take the system from any state that it starts at to any other state I want to push it to by injecting energy into one region? And well, obviously that's a really hard thing to do in general, but there are these beautiful results suggesting that in order to address that question quantitatively, you can use the smallest eigenvalue of the t-steps controllability Gramian, which is given here, right? So A is my adjacency matrix, and then B sub kappa is the vector that tells me which kappa regions I'm going to inject energy into. For brain networks, for this structural connectivity that I've been talking about, that smallest eigenvalue is extremely small. So it's 2.5 times 10 to the minus 23, which is within the precision of MATLAB, and therefore we cannot be confident that it is anything greater than 0, right? So this system is extremely hard to control, which is no surprise to any clinician that I've ever spoken to, right? The fact that psychiatric disease is still very present and real and not something we can solve very easily suggests that the system is very difficult to control. Mm-hmm.
What's the median or the largest eigenvalue? Because isn't the smallest eigenvalue out of context meaningless? Wouldn't you need to check for relative magnitude between the other eigenvalues and the smallest eigenvalue? Because I could always...
no, because the smallest eigenvalue is basically the ability to direct it to the most difficult, the state that's going to require the most energy, right?
So if I multiply my matrix by 10 to the minus 50,
well, no, so there is a normalization that's being performed, so you divide it by its largest eigenvalue to stabilize the system. Okay.
What's the size of A? Dimensions of A?
We study on the order of a couple hundred or 1,000 regions, so N is a couple hundred or 1,000.
I mean, generically, systems are always controllable. So 10 to the minus 23 shows.
They're not controllable if you can only inject energy into a single area. If you have all n regions as control points, then yes. But not if you have a single.
On a single node. If you take, they simply are generically controlled. It means if you take A randomly, which you don't have a random A, the system will be controllable. When it fails to be controllable, usually something quite drastic that you have, like missing an edge or something quite obvious. So I was just wondering if from your previous diagrams you could identify that.
No, there are no regions that are not actually connected inside of the network.
This might be related. So how robust is this in the sense that maybe if you don't want to control exactly, but you want to approximately control?
Right. So that's what I'm going to talk about next. Yeah. So one of the questions is it even relevant to be talking about absolute control of the system, or should we, especially if we're trying to ask questions about cognitive control, which is this ability to switch between different tasks or inhibit inappropriate behavior, that's not can I control the entire thing, that's a specific task, right? So what we'd like to do is to talk about specific control strategies and how they may be implemented in the brain. So there are 3 control strategies that have previously been suggested in the literature and that I think are very relevant for understanding the human brain. The first one is average controllability, and intuitively this is the ability to steer the system into many easily reachable states close by on the underlying energy landscape. Modal controllability is the ability to steer to a few very difficult to reach states far away on the underlying energy landscape. And then boundary controllability is the ability to steer the system into states where communities or modules inside of the network are either integrated with one another, communicating with one another, or separated from one another. So I want to give a little bit of math and intuition for all 3 of those ideas. So as I said, this is the model that we're working with, and here's the controllability Gramian that we're working with. The average controllability is equal to the trace of the inverse of the Gramian. So intuitively, that's basically sort of the impulse response of the network. And if you want to put that on in your head, and graphically speaking, what you're talking about is the ability to steer the system into local states which do not require massive amounts of energy to overcome barriers. The modal controllability, this is based on an eigenvector-eigenvalue decomposition of A. So we can let V sub j be the jth eigenvector of A with a particular eigenvalue given by lambda j. Then if the element Vij is small, then the jth mode is poorly controllable from node I. So then what we'd like to do is to define this summary measure, phi sub I, which is a scaled measure of the controllability of all possible n modes from region I. So that gives you an indication of basically we're looking for states that are particularly difficult for the system to attain. The boundary controllability this is actually a heuristic, so it does not have a neat definition that we can write down currently, although that's something that we're working on. The boundary controllability is the idea that if we have a hierarchically modular graph, we'd really like to understand which regions sit at the boundary between 2 modules, but consistently, independent of whether we're talking about large modules or small modules. So what we do is a hierarchical decomposition of the system into modules of different sizes, and we look for regions that are consistently at the boundary, whether we're talking about large communities or small communities. If a region is at a boundary between communities, whether we're talking about large or small, it will have very high boundary controllability. So where do these 3 types of controllers sit inside of the brain? So here's that picture that I showed you earlier, regions of the brain that are really important in cognitive control. And then here are these 3 types of controllability in each of those sectors. So this is average controllability first, modal controllability, and boundary controllability. If you just look in the default mode system, the preference is to have average controllers in this system. There are fewer modal or boundary controllers, although there are some of each type, right? So that's important. You have different kinds of controllers in each of these systems, but there's a preference for this average controller. In the green areas, the frontoparietal system, you have mostly modal controllers, but you have non-trivial representations of the other 2 as well. So it's not a perfectly clean segregation. And then for attentional regions, these have the largest amount of boundary controllers. So what does that mean intuitively? On this slide, what I want to give you is an intuition behind what these types of controllers might do for you in terms of what you do throughout your day. So I want to start with the blue regions, these default mode areas. They show white matter connectivity patterns that are predicted to effectively drive the brain to nearby states. Why is that interesting? Well, these regions, these default mode regions, are the ones that are very active when you're sitting and resting, not doing anything in particular, right? So these are often called your baseline areas, or your resting state, or your intrinsic state. I'm a physicist by training, so I call them the ground state. But the idea here is that these regions, which are very highly active when you're just resting, are the ones that are predicted to have the best capability of moving you to lots of local states nearby on your energy landscape. I would say, intuitively, that would be an optimal way of constructing a system, is to put it in its baseline in a place where the controllers are active that can push it to most of the places that it needs to go, right? Broadly speaking. The second point I want to make is these cognitive control areas. They show white matter patterns that are predicted to effectively drive the brain to distant states, far away on the energy landscape. So this is the blue line here, right? And again, these are the areas that are most important in helping you switch between different tasks. So solving the math problem and then talking to your grandmother or your child. So I would, that's sort of consistent with what cognitive neuroscientists have suggested that these regions do for several decades. But this gives a structural network reason for why that happens. And then attentional areas, so these are the areas that are really important in directing your attention to something that's in front of you or moving your attention to something new. And these are the areas that are predicted to effectively integrate and segregate different clusters inside of your brain. And that's very consistent with what psychologists know of these regions as being important for gating information between different areas. So what's interesting about this is that it suggests that the processes that each region of your brain does for you is can to some degree be predicted by the structural pattern of the network underneath. So this suggests some interesting questions for psychiatric disease. So I really like this paper from Randy Buckner's group at Harvard in Dialogues of Clinical Neuroscience, where he shows these same three networks, so the default mode, the control, and the attention network, and here's what they look like in healthy individuals. So you can definitely the strong modular structure, right? And here what you can see what it looks like in individuals who have psychosis. So the boundaries between the modules are very blurred. So there's an integration of these modules when there shouldn't be. And that suggests to me that there's probably alterations in the boundary controllability of these individuals. And that's something that we would really like to test in some ongoing data collection. Okay, so this suggests sort of a summary here. It suggests a network mechanism for cognitive control. And what's interesting about that is that most people, when they talk about cognitive control, will talk about what the processing units of your brain will do, so what the actual cell bodies are going to do inside of your brain, whereas this suggests that if you look at the structural connectivity, the actual network underneath, there's a lot about the function that you can predict just from the structure. All right, so that's a first suggestion that network control may be really important in cognitive control. But I'd like to ask whether there are a couple more lines of evidence that may help to bolster this story. The first type of evidence I'd like to talk about is how network control emerges in youth. So you might know that if you have a 2 or 3-year-old, their motor cortex and their visual cortex are actually very developed, right? So they can run, they can see almost as well as an adult. There are many things about them that are actually very, very well developed. However, cognitive control is something that doesn't actually fully develop until you're in your 20s. So when you ask the 2-year-old or 3-year-old child not to hit their friend because they're angry, that's something that's extremely hard for them, right? Much harder for them than running. They can run just fine, but they can't stop themselves from hitting their friend, even though they know it's not the right thing to do. So cognitive control is actually one of the processes that is the latest to develop in humans. It's also one of the processes that sets us wildly apart from many other organisms. So what we thought is that if cognitive control is something that's emerging in youth, particularly over adolescence, then we should see network controllability changing over that same time period too. So we use a really massive data collection effort called the Philadelphia Neurodevelopmental Cohort, and all of this data is actually online and freely available for anybody to use. So this was a NIH-funded study that was led by Raquel Gurr at the University of Pennsylvania in the Department of Psychiatry, and it sampled 10,000 children in the Philadelphia area. They have extensive cognitive phenotyping, meaning that we understand what cognitive abilities they had in spades versus not so much. And then they also had extensive genotyping as well, and all of that data is available too. Of that 10,000, 2,000 of them had neuroimaging data collected, which means that we have brain scans from 2,000 of those 10,000 children. And then what we did is that we looked at that imaging data, and we did a very stringent quality assurance process where we took out all of the children who had tumors, for example, or epilepsy, or anything that would make them not typically developing, because we were very interested in studying typically developing children. We kept the data, obviously, for everyone else, but they're not included in the data that I'm going to show you today. We're just looking at typically developing children. And then we also cut out any data that had a large amount of motion artifact. So particularly in magnetic resonance imaging, if there is a lot of motion artifact in the images, it's often very hard to be confident that the data is giving you a network that you can believe. So that brought us down to 882 individuals. And so here's a graph of the modal controllability per subject and the average controllability per subject. And here, each data point here is a person, one of those 882 children, between the ages of 8 years old and 22 years old. What you observe is that there's a positive correlation between these two statistics, indicating that people who had high average controllability also tended to have high modal controllability. We wanted to know whether...
Each person gave one data point?
One data point, yes. We wanted to know whether that was something that was consistent with simple null models. And what we did is that in the peach ellipse here, we have the 95% confidence cloud for a null model that preserves the node degree inside of the graph. And then in the pale yellow, we have the 95% confidence ellipse of the null model preserving node strength, because again, these were weighted graphs. And so what we see is a couple things. The first thing is that average and modal controllability are positively related to one another, but then we also see that the amount of controllability that's present in these children's brains is higher than we would anticipate in those two null models that retain either the degree distribution or the strength distribution. We also observed that actually both types of control increase with age. So as children age from the age of 8 years old to 22 years old, controllability of both types actually increases. And that's consistent with our hypothesis that as children are able to control their behavior more, the controllability of their network increases as well.
So what again do you mean by controllability when you quantify it, not as a binary notion? The controllability of 0.9 versus 0.95, what does it mean?
This is based on the equations that I gave earlier. So the average controllability is based on the trace of the inverse of the Gramian, and then the modal controllability was based on the eigenspectrum.
Trace of inverse of the Gramian, that's the control energy needed, right, on average?
That's right, yeah.
Understand.
But the next question that we had was, if you have this increase in controllability, is it that the 8-year-old's control points are different than the 22-year-old's control points? Or are they the same control points in an 8-year-old and a 22-year-old? It's just that the controllability increases in magnitude, right? So those are two different things that could have happened. It could be that the 8-year-old just uses completely different brain regions to enact control than the 22-year-old, or that we all use the same regions to enact control, but that the strength of that controllability changes as kids grow. And what we actually found was that the latter was true, that it's the same control points that are used in both the 8-year-old and the 22-year-old, but the amount of controllability increases with age. So here what you see, for example, is the average controllability of individual brain regions, and this is the degree to which that average controllability is correlated with age. So what you observe is that for regions that have, on average, high control, they increase in their controllability with age. So this is a positive correlation here. Whereas regions that have low amount of control decrease in their controllability with age. So what that indicates to us is basically that the distribution is changing, right? So in an 8-year-old, you have a relatively narrow distribution of controllability, and then as you go to the 22-year-old, it broadens. So the same regions are controllers, but they sort of increase on both sides. The regions that are most important in that changing controllability are located in the frontal cortex and in anterior cingulate, which are regions that are very important for this task switching. And in fact, we also found a correlation between the amount of controllability in a person's brain and their specific amount of cognitive abilities. So people who have higher controllability have better cognitive abilities than people with less controllability. Yeah.
Is this because the network evolves, it becomes more dense, or?
It changes in its structure.
Yeah.
Right. So it's not.
It doesn't become.
It doesn't become more dense. It just changes in the architecture. Yeah, but that's the next sort of question: is how do these changes map on to other graph statistics that we could calculate from the same network, right? What we see is that modularity increases, so you have stronger modules as you grow. The system is more segregated than it is early on in development. So I have a couple other stories to tell you, but I think given the time, I'm going to just end here and leave maybe some of these ideas to questions. In summary, what we did is that we talked about cognitive control as a flexible network process. We suggested that network control theory offers a framework in which to understand cognitive control, or how the brain actually controls its own behavior. We talked about developmental trajectories and how children grow in their controllability as they evolve from the age of 8 to the age of 22. We did not talk about preferred states or alterations in following mild traumatic brain injury from car accidents, but if you'd like, I'm happy to talk about that over the coffee break. And finally, I'd like to acknowledge the funding that has supported this work, the individuals in my lab, and I wanted to call out a few people. So this whole cohort here were extremely important in the data collection of the Philadelphia neurodevelopmental cohort and in posting all of that data online, which, as many of you know, is a really non-trivial effort. And so it's really fantastic that they are actually willing to share all of that with the world. Fabio Pasqualetti is my collaborator at UC Riverside in mechanical engineering, with whom I've been working on many of these ideas in control. And then the students who were important in the work that I showed you are Evelyn Tang, who is a postdoc in my lab. She did her degree in physics at MIT, Rick Betzel, who's a psychologist, and Shi Gu, who's an applied mathematician. And with that, I'd love to thank you for listening and take questions.
How much of this work was related to task-related fMRI, and how much was it resting state? So when you talk about controllability, do you mean controllability from some of the resting state aspects? Because I wasn't sure which. This was the structural one, of course.
Yes.
I understand that, but some of the work preceding this. Like, for example, here in the cognitive control network, so these were task-based, is that correct?
That's correct.
The visuomotor ones. But you use some of the notions from resting state later on, is that correct?
Oh, that's a great question. Okay, so this is all task-based data, and here when we build the network, it's a functional graph, meaning two regions are connected if they show similar activity patterns, right? Activity time series. For all of the controllability work, we're writing down a model of dynamics that occurs on top of the structure. So the data that goes into this is the structural connectivity, and we're, in some sense, predicting the resting state. But it's not explicitly in the model. We're predicting it. And then when I talked about the resting state, I was talking about regions in particular that are known to be active at rest. What we're looking at is the structural connections that are present in those regions and from those regions to other regions. So using diffusion imaging.
How do you
measure similarity of time series? Because there are very different
Yeah.
ways of measuring similarity if you have two time series.
Yeah. What we usually do is that first we use a wavelet decomposition to separate out a frequency band that is less impacted by cardiac artifacts and by respiratory artifacts. And so after we do the wavelet decomposition, then we look at coherence of the wavelet coefficients. Yeah.
So you basically do some kind of thresholding, and then you are looking at. Are you looking at the
We don't do
size of
we don't do
coefficients
any
or-
thresholding.
Yeah. Are you looking at the size of coefficients, or whether it is present or not, or what do you do?
We're looking at the coherence of the wavelet coefficients.
Coherence meaning.
So it's basically we're looking at so coherence is actually estimated for each frequency. Well, the similarity in the power spectrum for each frequency range. So what we're doing is that we're taking the magnitude squared coherence over frequency and time.
Each resolution level.
Say that again.
Energy at each resolution level. Did I understand you correctly?
The energetic resolution.
Energy at each resolution.
Yes,
Different
that's right. That's
resolution.
right. Yes,
So
exactly.
energy at each
Yes,
resolution.
mhm.
Okay.
Yep.
I mean,
Yep.
it's just different terminology. I was
Right.
just trying to figure out what stands for coherence.
Okay, so, okay, last question then.
Very quick one. How do you choose the time window to construct the network?
That is a great question, and it significantly depends on the time resolution of your data. For fMRI data, we have a study that just came out in NeuroImage this year, where we look at many different time window lengths and ask how consistent are the results if we image people on different days, for example. So we're looking at a question of reliability. And for that, we suggest that going any smaller than about 30 seconds, you have a lot less, you have decreased statistical power and increased uncertainty, whereas 30 seconds and above is relatively reliable across multiple measurements.
From 30 seconds, how much larger can you go that it still is meaningful?
You can go very large. So in fact, if you take 6 minutes of data and then you take the next 6 minutes, you're still going to see changes. And largely that's because there are many different things that actually impact what our brain is doing. Whether we've eaten and had enough coffee, you're probably all feeling that, you need some more coffee, that actually changes the pattern of functional connectivity in your head, and so that's going to change over pretty long time scales, right? Another item that really changes the pattern is the mood of the person. So if you're actually in a happier mood versus a sadder mood, the patterns change as well. So that changes on the time scale, usually of days. For people, although there are more transient effects too, but the really salient one is timescales over days. So I wouldn't necessarily say there is an upper end or upper limit.
Probably a good time for a coffee break.
Yes, I agree.
We're back here at 4 o'clock, and let's thank Danielle again.